Hello everyone, I'm Johannes, the convener of the Convergence Science Network, and on behalf of its sponsors, I'm delighted to welcome you to this event where we share advances in the biomedical sciences. This evening, I'm pleased to have a recognised scientific expert in the field joining us to share the science behind cell-based meat, sometimes also referred to as cultured or lab-grown meat. But first, let me welcome Bianca. Bianca is the founder and director of Cellular Agriculture Australia. Bianca will introduce our guest speaker and moderate the Q&A after the talk, and you'll see me a little later on. So over to you, Bianca.
Hi everyone, and thank you Luan and the Convergence Science Network for hosting this event to inform all of us, all of the community about all of the advances in biomedical sciences that impact our health and our environment. My name is Bianca Lay, and I'm the Director of Cellular Agriculture Australia. We're the leading nonprofit organisation dedicated to promoting and accelerating the research in this emerging field of cellular agriculture in Australia. So I'll be moderating for today, and I wanted to start by acknowledging that I'm moderating this webinar from the lands of the Wurundjeri people of the Kulin Nation, and also pay my respects to their elders. Considering the topic of this seminar, I think it's important to note that the Wurundjeri people were the original scientists and engineers and farmers of our land. There is now a lot of evidence to show that for tens of thousands of years, the Wurundjeri people successfully managed the territories and the lands around the Merri Creek here in Melbourne by engineering biodegradable tools and equipment that provided renewable sources of food and clothing for their people. And this was all done in a very sustainable way without any disruption to their ecosystem. And so here we are today in a state of the world where sustainable farming is now more important than ever. And some of you in the audience are probably tuning into this talk because you're just as passionate as I am about saving our planet, our people, and our animals by innovating our food systems. So it gives me great pleasure to introduce our guest lecturer for today, Professor Johannes Coutre. Johannes is one of those great examples of a scientist who has an outstanding career, not just because he's been successful in both academia and industry, but also because of his work in actually uniting the private and public research sectors together. Johannes started by getting his PhD in biophysics at the Max Planck Institute of Nutrition Physiology in Germany. He then went to the Howard Hughes Medical Institute at UCLA to research molecular mechanisms of membrane transport. In 2000, he then made the move into industry, where he built a research program on taste physiology at the Nestlé Research Center in Lausanne, Switzerland. Johannes has since developed various large-scale global research programs, which involved professorships at the University of Tokyo and Imperial College London. In 2019, so right before the pandemic hit, Johannes made the big move to Australia to establish the very first academic lab dedicated entirely to cellular agriculture research in Australia at the University of New South Wales. And just last year, Johannes was selected to be on the advisory board for Cellular Agriculture Australia. So today you'll be hearing a 40-minute presentation from Johannes about all the different hats he's worn and currently wears as a scientist, as well as a deep dive into what cellular agriculture is and some of the technical challenges the field is currently tackling. So without further ado, thank you so much, Johannes, for joining us.
And thank you, Bianca, for that amazing introduction. That was the most comprehensive introduction I've ever gotten. Very nice. Thank you for having me. Thank you for the organization for having me. So what I was thinking, what I will do is to talk about two big parts. And you see the title indicates that we're talking from healthy ingredients, which is my past life, to cellular agriculture, which is my ongoing and hopefully future life. Which is sort of a story of the field almost, where we go from ingredient research in the nutrition and food sciences to really looking more at meals, at dishes, at diets, to integrate the whole thing. In a way, it's the story of the field. It's also the story of my life in a way. So it's interesting. And what I thought I will do is I will tell you a little bit of what I started when I joined Nestle 20 years ago to talk about taste. Hopefully, we will manage to close this circle at the end of the day, to come back to taste. After all, taste is one of the key features of food, right? I will then talk about a little bit about gut tasting, TRP channels, transient receptor potential channels, spices, and metabolic health. All of these are ingredient questions to then really put the second half of the talk really into the evolution of 21st century food systems. And that is supposed to be cellular agriculture, cultivated meat, cultured meat. There are many names for this, and we will be getting there. Okay, and I will get back to that outline each time we change gears. So first, very quickly, when people talk about taste, they very often mean flavor, actually. And flavor is taste plus smell. So what you can do is you can, and that's a very simple slide for the students, which I still think is good to show you here also. You can take a nose clip your nose, blindfold your eyes so that you cannot smell and you cannot see. And if you then will put and ingest and eat gratings of either an onion or an apple, you basically will not be able to distinguish the two of them because you are just left with the pure taste, which is the gustatory component. You have blocked all of the irritating smell of the onion with the nose clip, and you cannot see what you're eating. So they will both taste the same. And flavor is really a very complex and complicated thing. And again, you can do this and try this, for example, with drinking a cup of coffee with a clipped nose. You will see you have a totally different experience. The pathways, and this is really just the introduction into the entire subject. The pathways we know, and this is work that goes back many years, but started to really pick up with the identification and sequencing of the human genome. We know there are 5 different gustatory taste modalities. There is sweet, sour, salty, bitter, and umami, which is the taste of glutamate, of MSG. And we know there is 2 olfactory pathways, which are basically the same. One is just the direct nasal pathway, and then there's a retronasal pathway. And again, once you clip your nose, you block them both, and you are left with your gustatory sensation, which is conveyed through the either chorda tympani nerve here or the glossopharyngeal nerve, which synapse with their first synapse of the nucleus of the solitary tract before they integrate in the brain with other taste modalities. There is, however, and this is relevant for the second part of what I will show you, there's another system which is the chemesthetic system, which is the system to detect irritation and spiciness, and that is conveyed and taken care of by the trigeminal nerve, which is called trigeminal because of its 3 roots. One going into the eye, the other in the nasal cavity, and the third one into the mouth. Again, being responsible for the detection of spices, and we will get to that in a second, or not in a second, maybe in a couple of minutes, 5 slides or so, when I talk about our spice work that we had done in the past. Now, if we zoom in, going back to classical taste, the 5 gustatory tastes of sweet, bitter, sour, salty, and umami. If we look onto our tongue, and this we can do in the morning in front of a mirror, we see these dimples, which are papillae. With a basically naked eye, we can see those already, which are so-called taste papillae. Each of those well, actually not all of them, but most of them are decorated at their surface with so-called taste buds. And it is the taste bud that is the elementary organ of gustatory taste perception. These are associations of about 100 so-called taste cells. Do you hear me? There was a funny echo. These are associations. Yes? Hello? All good? These taste buds are associations of about 100 or so neuroepithelial derivatives, which are taste cells with their apical end oriented to the surface of the tongue, the surface of the tongue. So these taste buds, they are made like a lemon or like an onion with the apical end exposed to the surface of the tongue. This is where food is passing by, and at their basolateral end, they have their gustatory afferent axons, which are those that project into the nucleus of the solitary tract. Okay, if we zoom in even further at the apical end of the taste receptors of the taste cells, you find expressed taste receptors, and this is really, I need to point that out, work that has been driven forward predominantly by Charles Zucker, many other important scientists as well, but Charles was really the one who sort of put the pace into the field and who really dedicated always at least 2 Cell to Nature papers to each of those taste modalities. So what you see here is clearly that different taste modalities are activated and triggered at the apical end of the taste cells by specific taste receptors. Different taste modalities use different taste receptors, and they can be used, and that is what big food companies are doing, smaller ones as well. They can be used, those taste receptors, to discover novel flavoring molecules. So this is very much a pharmaceutical approach. This is a pharmaceutical approach where you express those receptors in a heterologous expression system in vitro, and you can use them then to find umami tasting molecules, sweet tasting molecules, and what have you. Now, the umami taste receptor is a heterodimer composed of 2 of these big GPCRs, one of them the T1R3, and this heterodimer is sensitive to both glutamate and nucleotides, which is the taste of umami, and we will hear a bit about that in a second. The sweet taste receptor, again, a heterodimer of big N-terminal C-class GPCRs with a big N-terminal binding site that builds up this venous flytrap ligand binding domain to form a binding site for various types of sweeteners and sweet molecules, such as sugars, artificial sweeteners, certain amino acids, glycine, you name it, also proteins, which is different to the case of bitter taste receptors. And bitter taste receptors are small GPCRs with a binding site with a very small N-terminus, with a binding site in the low dielectric of the membrane. But we have many, and that makes sense because they warn us, or they have a role to warn us, or the activation of those bitter taste receptors plays a role in warning us in case we ingest toxic compounds. What is interesting in this whole case, in this whole scenario here, is something that you can see in vitro, but that you can also see actually with people, is that certain molecules like saccharin in the millimolar concentration range start to activate bitter taste receptors. Now, these molecules in micromolar concentrations activate sweet taste receptors, and only if you up the concentration, they start to convey a bitter signal, which is something you clearly can show in vitro, but it sort of resonates and explains why there are many consumers out there who perceive an unpleasant or unpleasant side taste upon ingestion of artificial sweeteners in other products. And that's very interesting because it tells you can really use these assays to mimic the perception of foods. There is sodium taste, which is the taste of salt. Again, it's an ENaC channel. Interesting, the ENaC channel has not been discovered by Charles. The ENaC channel has been discovered down the road of Nestlé Research Center by the Rossier Group at University of Lausanne. And we have an even more complex detection system for sour taste. And I will, again, these are channels and not GPCRs, and I will not talk about those. What I wanted to show, and again, I hope we will get back to that. What I wanted to show to you is both. You can do this on the molecular, but also on the sensory level. Hallmark of umami taste. And umami is got a lot of controversial press in the early 2000s. I think it's a bit over the whole story with Chinese restaurant syndrome and so on. But basically, you have an EC50 in the millimolar range, in the lower millimolar range, which with a sigmoidal dose response curve, like it is shown here, and just upon addition of very small amounts of nucleotide, you can shift this entire dose response curve to micromolar ranges. Now that is clearly the taste of meat. There is no other sort of compound which is highly packed with amino acids and with nucleotides like meat cells are. And I think it is an interesting thought to keep in mind in the discussion we are going to embark now on, that we as humans do seem to have something that might be a taste of meat. Okay, and as we now move on, just the last two slides in this section, just for completeness, there has been a long-standing discussion about taste. Are we talking about something where every taste cell in a taste bud can do everything somehow more or less good? Or are we talking about a situation where we have a labeled line and where each and every taste cell in a taste bud is responsible and only responsible for one and only one taste modality, such as this red one here for sweet, this light blue one for salty, and so on and so forth. And it has been, again, Charles Zucker who really showed in a series of beautiful and very elegantly conducted knockout experiments, and again in about 24 of Cell and Nature papers, that we are talking about a labeled line both at the periphery and as we go higher into cortical areas as well. Lately there has been literature out there which sort of puts all of this again a little bit into doubt, but that's science as science goes, and it's an exciting field to be in, because you can see that a perception can be really immediately treated and handled in the laboratory. If we move on, and then we switch gears, signal transduction, I just want to point out, is for all these taste modalities on the GPCR and sweet, bitter, and umami, driven the usual way with mobilization of intracellular calcium. Please have a look at this TRPM5 channel, which is a taste signal transduction element, which can be also used just as a marker or a tracer. And here you see the ionic tastes, which just lead to depolarization of the membrane and release of neurotransmitter at the basolateral end. Okay, and that is a paper done by a good old friend, Sid Simon, in the field. Now, I would like to switch, and this really reflects a little bit how things had been happening back in the day. Those were the early 2000s in Lausanne, to what is gut tasting, or what we call gut tasting. And the story here is really that basically, once you have all of these primers, probes, and plasmids, most notably primers in the lab, you can also interrogate other tissues. And that is something that had also been done by other groups. And it does have relevance now, what I'm showing to you, to food and to food ingestion, food perception, and food metabolism. The Drenkan group in Germany in the late 90s was able to show expression of alpha-gastusin in the rodent rat gut, like it is shown here. It was then at my alma mater, Rosengurt, the Rosengurt group, after I had left, group who had shown, again, this is a bit debated, but still expression of T2R, of bitter taste receptors in the rat gut. And we're talking about this species up here. And finally, our group, together with Carole Besançon, Sami Damak, who had just joined us from Monel, and a few others, we could then first show that, TRPM5 and subsequently the T1Rs are expressed all the way through. So the receptors for sweet taste and umami taste are expressed all the way through in the rodent, in the mouse gut, which altogether with the findings of the colleagues really is a spectacular observation. Because why, you can ask yourself, why would a system evolve that has taste in the mouth and can taste food, and why would we use taste principles in the gut. And I think the early working hypothesis, or I know the early working hypothesis had been there will be a role in hunger, there will be a role in satiety, there will be a role in gut motility, and I guess it's fair to say that all of those are involved. There is a number of interesting observations. One would be, for example, that sweet taste receptors, the T1R2 and T1R3, and that comes from the Bob Margolski group, who then went to Monell, that the sweet taste receptor in the gut is involved with sensing levels of sweetness in order to recruit SGLT, which is the principal sugar transporter. So you have a sensor, and the sensor is responsible in the gut to open up the floodgates, if you will, in order to let the sugar in, which is metabolically super important. Other work, again, by Charles, but by many others, shows that these gastrointestinal tasting systems really have a role to play in gut sensing, but also in the gut-brain axis. And I'm not going into this domain as well, into the gut-brain axis. That can be another talk. Instead, we're talking about TRP channels. Just quickly, I would like to show you a little bit, because we are still in the healthy ingredients part of the talk. And as all of these taste receptors had been identified, in parallel, the Pataputian group in San Diego, the David Julius group in San Francisco, many other groups had shown that those transient receptor potential channels work as the sensors for spices. And that is interesting, because spices per se do not really have a nutritional attribution and do not really have a nutritional role as far as we think, or as far as we had thought. At least there is not much about the nutritional role of spices. However, they all have some anecdotal, and certainly had some anecdotal benefits, such as antibacterial, anti-inflammatory. You see it for yourself: antibacterial, cinnamon, anti-inflammatory, tissue protection, blood sugar reduction. I will show you in a second something about cinnamon. Nutmeg is an interesting one, has also a very strong psychoactive component. Basil, good source of antioxidants, and so on and so forth. So what we thought really is that with the arrival of all of these receptor systems that can be manipulated in the laboratory and that can be used in the laboratory, we have a good tool to study the role and to put some science onto those anecdotal benefits. And just to illustrate that a bit further, I mean, if you think pharma has complicated and complex molecules, here is just a quick overview that shows you that basically spices and foods are complicated, complex molecules as well. Of course, here look at this ginsenoside, super complicated steroid. Look at all of the saffron. So the simplest one is possibly cinnamaldehyde. I will show you a bit more about cinnamaldehyde. And again, just the point to make that there is reason to believe that foods and spices, and I have a slide in 3 slides from now that makes that point even more. There's reason to believe that foods and spices can have some pharmacological properties. So just to show you the TRP channels, those are broadly tuned cation channels, that they are interesting animals in a way. And the most dominant and famous one, I guess, is TRPV1, transient receptor potential channel vanilloid 1, which on the one hand is a chemical sensor. It opens up upon binding of capsaicin, which is the fundamental hot and spicy molecules of chili peppers. But it also opens up when you increase temperature, which means if you feel hot after eating spicy food, in a physiological way, even though you might not raise your temperature, in a physiological way, you actually are feeling heat. Same thing on the other end of the spectrum is the TRPMA channel, which is a channel that is activated and opens up and gated by menthol, but also by lowering the temperature, as you see here. Okay, and our objective had been because by then in 2011 many groups had shown already that the TRP channels are really ubiquitously expressed all over the place. We even showed and have a publication out there also that in rodent brains, so TRP channels are all over the place. and are associated, you will not be able to read this, are associated with a lot of diseases and malaises. So we thought if we have receptor systems for spices, which in the body do have a health-related role to play, we possibly could use something like a perceived benefit associating spices with health. That was the idea, and we did this for a number of systems. Just to show you one, and I told you there's another slide. I forgot about that. The underlying, and I just like this figure, the underlying mindset we are following, and I guess that is something which will come back also for the second part of my talk. The underlying mindset for what I will of what we have been doing is clearly that food can or has a role to play as a building block. It put this 100-kilogram guy together. Food has a role to play as a source of energy, but food most likely also has a role to play as a signaling agent of some sort, which gives signals into our body. And what we did is we started to look at cinnamaldehyde from cinnamon, the key molecule. We found expression of the principal trip channels in the mouse gut, but we also found that it is co-expressed in ghrelin-secreting cells. So cinnamaldehyde triggers ghrelin-secreting cells in a way that addition of the ligand of cinnamaldehyde leads to a reduction of ghrelin, and we call this then our little cellular endocrinology. Also has an impact on the expression of TRPA1 itself and the insulin receptor. And I hurry up a little bit because I see time is flying. Interestingly, if you do mouse experiments, and we did subsequently do this also in a human clinical study, you can show that in rodent studies you can induce in high-fat diet fat animals that you can induce a 10% loss of body weight at the same amount of food intake just by the addition of cinnamaldehyde. I will not go any further here, and the point is really there are ingredients that do have a role to play in our individual health, and there's tons of more work that can be shown, presented. That will be another talk, I suppose. Now the key chapter is really to present here and to put that into the context of a change of nutrition science into looking at diets, looking at meals, but also looking at our planet and looking at sustainability in the bigger picture, which is certainly what I signed up for, but is also what the entire field is pursuing somehow. And to give you reasons for doing this, I show you here very quickly a few points why we do have issues on Earth. We do have issues on this planet. We have issues with aging population. 20% will be over 65 by 2050. Malnutrition is a big issue, and we're not talking only about, well, we are certainly talking about over 800 million hungry people. We are talking about 2 billion. We are talking about 2 billion seriously malnourished people in poor countries, but we are also talking about all of those that are malnourished and that have overweight, obesity. All of this is a big and ever-growing number, and we have a growing issue with allergies, food allergies, and food intolerances. So that's not all. There is more, and you will see that actually our food system is culprit in many things. What is a food system? A food system is shown here, where you go from the beginning to the end. You see here our green agriculture, which is all of the growing of plant material. You have our livestock agriculture, which again speaks for itself, I would say. You have the processing industry, or again, this can also just be a kitchen. You have distribution of food, and you then have purchasing and ingestion and consumption of food. That is a food system. And what I show you here with those red stars, and I do use that slide always as a pitch to justify what we're trying to do here. What I try to indicate with those stars are actually problems. So we do have in the green agriculture part of the food system, we have an issue and problems with arable land use, with water balance. We have a lot of problems when it comes to livestock with regard to food security, food safety, animal welfare, environment, you name it, and there's more. Food waste is all over the place. You probably all know that we are looking at about 30% food that is lost. Packaging and plastic, I will not talk about that, another big problem, especially when it comes to microplastics. There is more, there's the growing food population, there's the demographics, which makes us or necessitates that we increase the available calories by 70%. And if we now only talk about livestock and protein and food, livestock and protein supply, it is very clear that conventional livestock systems have reached their limits. And this is talking about freshwater used to grow livestock. It is about greenhouse gas, very big issue, and much more so in the livestock agricultural sector than it is in aviation, for example, which is only 2%. The land used for growing feed is 33%, and the land used for having those animals and all of this livestock, 45%. We have, in addition, a big health-related issue, and we all have seen that, I believe, right now with COVID. There is more. There's hormones, antibiotics, microplastics. There is what I call ethical correctness when it comes to the limits of our conventional livestock systems, and that is animal welfare. 75 billion animals, souls, are being destroyed and slaughtered per year. And that is not counting fish, because fish goes by weight. And that is not saying that all of this is the result of factory farming, but factory farming has a big role to play. And what it tells me is something is going wrong one way or another. And I actually like to eat a good steak. I have to say that, like Mark Post also would say at the beginning of his talk. So in addition, another problem is loss of biodiversity. We have economical impact, you name it. So we do have a problem with our livestock systems. And there is a beautiful listing of about 90 reasons to consider cellular agriculture, and I would agree to at least 75 of them. And I guess those slides will be available at some points, and you can then look that up for yourself. Now, there are ways to address alternative proteins to relieve our food systems a little bit and to push back the burden on our livestock systems. And again, we are talking a lot of plants, single cell protein, algae, spirulina. There is a lot coming from that, and I will go more now into cellular agriculture. I just don't want to omit that there is also big movements to have more insects on our plates to provide us with protein derived from insects. And you probably have heard about that. Now, the next slide is probably really the most important slide in the whole presentation. And I hope I get that across well. That's a slide from the Rethink X report 2019. And the point it makes is, and this is really the very, very big picture. We often get caught up in small pictures or in small problems like regulatory, VC money, grant writing, these types of things. But there is a very big picture to make here, which I'm trying to explain, is that food production traditionally is dependent upon domestication of both, of micro and macroorganisms, as you can see here. Where all of our yogurt, cheese, kimchi, sauerkraut production is dependent on bacteria. Fungi, we have our beer made by those. We like to eat shiitake mushrooms. We have algae in various forms that contribute to our food. And in what is called in the report, the pre-domestication, we started to enjoy consuming plants and consuming animals. Which certainly sort of solidified itself in the first domestication, which took tens of thousands of years. Again, we are talking about events in human evolution. We're talking about human evolution here and historical events in a way. But you see, we still have open gaps here, and we still do not manage, but we are trying to make sense of the, as Winston Churchill called it, absurdity of sort of domesticating the entire animal. And that is something that holds great promise on what I would say really on a historical, on a human evolutionary, social evolution level to get this done with. And again, if you look at this, it will then have the value of really talking. It's more than Haber-Bosch. It's more than, maybe it has the value of using fire in human evolution, more than inventing the fridge, and it's certainly also more than the Green Revolution. So if we get this done right, I think as a human species we're onto the right track. That was the most important slide. Now, how is all of this done? How is all of this done? And that is here from a slide from Elliot Swartz and Deepak Chowdhury, which I modified a little bit. Cultivated meat, when we talk about cultured and clean or cultivated meat, which is sort of a subsection of the entire cellular agriculture effort, we try to pull a biopsy from an animal. We then throw it into a bioreactor of some sort, let it grow, proliferate, develop, differentiate, and put it eventually or not onto some sort of scaffold and substrate in order to have materials that are edible, like a steak. And I actually, the part I deleted from that figure was an Erlenmeyer with a piece of steak in there, because that's sort of, I did not like that. The issues here, and everybody knows that, everybody in the field knows the key issues have been, and I think still are, medium optimization and serum replacement. You have to take out all of the FCS, because you cannot claim to have animal-free material if you need animal material to make it grow. And the scaling and cost. That, and in a way, if you look at it carefully, scaling and cost are the same. So scaling and cost is really the key issue, and we hear many things, and we will have a discussion just after this. We hear many things. I still think the jury is out how this will evolve, and clearly we are just trying our best as well. And if you want to run a successful program on cellular agriculture, you need to do a lot. It takes not only good science, upstream science, and many years of science, and the science done by many groups and departments. You need to do tissue culture, serum replacement, antibiotic-free, scaffolding. You want to maybe put in fat cells into your product. You need to maybe carry some iron, so blood might be interesting. You need to take care of the organoleptics. I told you we have a lot of know-how here about organoleptics. We are building, in fact, a big sensory activity in an academic setting in Australia. We have to consider taste, aroma, texture. If you want to then build your technology further and scale, you need to think about bioreactors. And there's a lot of talk about bioreactors, but it is clear also that we need to first get the science straight, and the science informs then the bioreactor design. Supposedly UNSW is running the biggest engineering operation in the Southern Hemisphere, which is good, so I'm in the right place, and we're trying to get this off the ground. Again, scale and cost are key issues. If you run or if you try to develop sustainable materials, you don't want to package it with a lousy shrink-wrap plastic. So again, here that is something to keep in our minds. And very importantly, and again, this is something which might have gone wrong with GMO, and which might have gone wrong with other things. You need to educate, grow your consumers. You need to ensure and reassure consumers. You need to have regulatory approval for a successful deployment of these materials. You need to know your supply chains up front. You need to engage with farmers, and you need to make sure the safety is taken care of. Okay, now I think I'm still okay in time. I am over time already. Apologies about that. I show you what we're trying to do in the lab. And our objective is to disrupt factory farming. Yes, but to empower farmers with new technology, while keeping traditional supply and distribution channels. So we want to produce meat. We want to produce meat. Somebody called that legacy meat. We want to produce meat which is the same meat, which is not plant-based meat. We want to extract and isolate the process that is happening in the animal, and we want to feed that process with the same stuff the animal is eating, i.e. with grass or with hay. We want to then run it through some biotransformation process before we put it into bioreactors, which then give us that material. Just to remind you, meat is, when we say meat, usually we're talking about muscle. Why muscle? Because it's tasty and it's a good and high source of protein. And to continue showing you what we are doing, the idea here, you have seen that already, is to take a biopsy from muscle tissue while keeping that animal alive, to cultivate that in a tissue culture setup. And we have fancy equipment, and we will be getting even more fancy equipment to refine the process, to think of products. And we don't think it's always a good idea to copy or mimic existing products. Why not thinking of something entirely new? And why on earth do we need to mimic a sausage? Nobody knows, and we see that in the field. And to feed that process by using as a carbon source and as a source, again, the traditional supply chains, possibly even food waste, to run that through a dedicated setup of biotransformation, which then will result in media that we can feed into this tissue here. Now I'm going to show you some results, and this is preliminary and is being written up and will be published anytime soon. And I hope the authors are sitting in the talk. And what you see here is fermented plant extract from plant material grown somewhere in Australia outside. So this is a fermented plant extract at different levels of purity and purification. I will not go into further detail. You see, it's also comes then depending on the purity. We're having different volumes of that material, and we are and have been eager. That was possibly the only good thing about COVID in 2020. We had a lot of time in the lab and didn't have anything else to do. So we thought we put that material onto myoblast cells, and much to our sort of, well, not totally unexpectedly, but actually that material is toxic. That material is toxic. The fermented plant extract per se does not work. However, if we go ahead and treat it further in a purely biological way, in a simple and scarily simple way, and what we call LOPO number 9, so that is love potion number 9. We do love potion number 9 to our plant extract. All of a sudden, something happens. Either toxic activity is taken out or some benefit is created. We don't know, and we will investigate. That all has been done in sheep myoblast cells. And to quantify that a bit better, what you see here is the situation in sheep, and here's the situation in a rabbit. The red is, in one hand, our goal when it comes to the simple numbers of the cells, but then again, it's not our goal because we're using FCS. You see the fermented plant extract per se at increasing concentration is toxic, and if we do LOPO number 9, we at least in the sheep case eliminate toxicity, and in the rabbit case we seem to be getting it very close to what we want to achieve with the FCS scenario. Now that is very encouraging for us. We are pursuing this further on a highly multiplexed way, and we're trying to understand what is going on and what needs to be done to get this even better at an certainly low cost, highly affordable way, and clearly in the framework of existing supply chains. So that is that. Now the last slide, and then I'm at the end, will be talking about our efforts to dimensionalize, to put really all of this also onto a scaffold, because all of these efforts so far in all laboratories worldwide are resulting in some funny looking slime. And what we have, we got hold of specific edible polymer material shown here. We are using a lot of scanning electron microscopy and transmission electron microscopy. What you see here is basically the edible polymer material that we are using in order to grow myoblast cells. And if you do that under certain conditions, what you get is these things here. You're also getting scenarios of these blobs here, which are actually the myosatellites that we're feeding in there under specific conditions. But what was really amazing was for us to see that we get attachment, number one. So the myoblast cells do adhere to the material, which was not evident in the first place. But even more exciting, the myoblast cells do begin to differentiate into spindle-shaped myofibers. You could even hallucinate or daydream about maybe a sarcomeric structure here or not. I don't know. This is not the best picture here. I noticed that too late. But what we do see is in our transmission electron microscopy that material really forms attachment sites. So cellular material like this big one here forms attachment sites with the polymeric material, and that is super encouraging and made us very happy. So that was my last serious slide. I want to make some advertisement for my own case, which is to really show you. We just published with the leaders, with the best scientists globally in the field across the planet, a huge paper, Goals in Nutrition Science 2020-25 in Frontiers in Nutrition. Please have a look at this. We also talk about cultured meat, and then the really last slide shows you some of my collaborators over the years in all of this. Nestle Research Center in Lausanne, the CHUV in Lausanne, which is not relevant for this one. For the work I had shown you, Sami Damak, Susanna Camacho, Stephanie Mischlik, and Celine Riera clearly had a key role to play. And here at UNSW, we get material from Bill Walsh. Mark Ripshin is and has been and is a rare find in the tissue culture domain, and I'm happy to have him on board. Julian Cox is always helpful with discussions. Joe Richmond is doing all of the electron microscopy, and James is just getting his feet wet in the laboratory to start with more significant media preparation. Thank you.
Thank you so much, Johannes. There's a lot of exciting research happening in the space in general, but it's really exciting to see, some of the world first work you're doing right here in our own country. So as a taste and nutrition expert yourself, you've shown us that there's still a lot more research that needs to be done to really understand the role of these different flavors and molecules in not only how we perceive food, but also how we digest food. I thought that was really cool. Cultivated meat itself is not available anywhere in the world at the moment, except in Singapore. So I'm sure no one in this audience has tasted cultivated meat yet. I have two questions. One, have you personally tasted cultivated meat? And two, what is the most efficient way, or how would you approach trying to maximize the flavor of cultivated meat? Would it be modifying the cells themselves, or playing around with the muscle cell to fat cell ratio, or even playing around with the scaffolds? How would you approach that?
Honestly, think if you can scale the biomass, if you have biomass available, it's all about having the biomass at hand. And we know from decades of food science, right, and food science engineering and food science technology, we do know that there are many ways to improve and change and alter all of the parameters of food. So we can put in texture, we can put in taste. I think I would, in the first round, I would probably really go for maybe roasting things or toasting or whatever, trying to get some Maillard reaction going, the usual taste molecules from spices to even salt to add. And to answer your question, I have not myself tasted any cultivated meat. But there are groups out there who certainly have done that, and I think the very initial perception of that stuff is a bit to trying to be objective. It's a bit it's not very impressive, I would say. I don't want to say dull, but I do say dull. So it's a bit boring, and it needs some treatment to enhance taste. I'm sure, but again, and taste and texture, I'm sure will be given, for example, also through our scaffold or through anybody's scaffold for that matter. And again, with the traditional ways, with some culinary art, there is so much that can be done that at this point, I think key is really to produce the biomass at scale at an acceptable cost. That is really the critical bottleneck right now.
That makes sense. And some of those research experiments you mentioned, like roasting and the Maillard reaction, I'd love to be a taste tester for that. So I'm sure a lot of our audience members will agree. You've got this really interesting career pathway where you've moved to Australia right before a pandemic. And I just think that's really fascinating that you've built this entirely new lab from scratch during that pandemic. And we've got a audience member's question that asks, can Australia be a major player in cell cultured meat? And do we have any advantages over other countries?
Yes, and yes. And I hope that my little presentation has a little contribution to make. That's why I came here. Look, that's at the end of the day why I came here. It's just, I mean, it's a beautiful country. It's a huge country. It's a country of abundance. It's a country of abundance in agricultural and natural resources, sunlight. So why should that? And that's even the sort of strategic incentive of the faculty when they recruited me, and we had discussions over a year. So I think Australia, if we in Australia get our act together the right way, then definitely we have a huge opportunity for those reasons. I can maybe say a few more things. There is a huge, strong and long-standing cultural heritage of high-quality food, right? We have that. We have a pride here in our technology and R&D. So I believe, yes, this can be done. And we have one big country that is all within one big legislation. So also the regulatory framework, which is discussion that needs to be held, the regulatory framework permitting, we can do that. Yes.
Couldn't agree more, Johannes. We've got, some of the most world-class, world-leading experts in regenerative medicine and cell biology research, as well as incredible animal agriculture sector with a great reputation around the world. So really strong export and trade agreements with a lot of our neighbouring countries. So yes, I completely agree. Australia is a great place to really build this cellular agriculture sector. You mentioned that cellular agriculture is more sustainable compared to animal agriculture in many ways, but exactly how effective is it compared to animal agriculture in terms of environmental pollution and carbon emission?
Honestly, I don't know, because many of those things we're talking about have not at all been established at the scale that we are running our current food systems. So it will be hard to make any predictions. I do remember there has been a paper somewhere, and that brings back again this Australia advantage. There has been a paper somewhere showing that it's many aspects are very feasible to the reduction in water waste, because things are becoming more manageable if we are looking into big bioreactors, right? And this will take a long time. Still, one point in that paper was that the energy costs might be going through the roof, and that is something we need to anticipate. We need to be aware of, and we should also not lie to ourselves that cellular agriculture will come for free. That's not going to happen, and it will be a tightly knit balance sheet with traditional and cellular agriculture on both sides, if you want to do the math here.
Yes, definitely a lot more research that needs to be done in that area. You mentioned that we have to take a biopsy from the animal. Does that biopsy harm the animal in any way, and is it something that we have to do frequently?
So I do recommend watch the movie. If we just did this today with the students, you can Google the Josh Tetrick's company, Eat Just, and then you Google Eat Just. You search for a video, and you Google, you put maybe in cultured meat and Ian and chicken. So what you see here in terms of, and it's a beautiful movie. It's such a well done movie. It's amazing. And what you see here is that a biopsy possibly might even be just a feather that naturally fell off that chicken. So that is sort of the totally zero pain end of the spectrum. And I guess you can, if you then go into maybe a bit more slightly, it is invasive, or it could be invasive to take a muscle biopsy, right? But you need only very small amounts, and that is something that we had shown that works. You don't need to take the life of the animal, very clearly, and the animal will not even be limping afterwards or anything. It will just be a happy animal which might have an itch in his rear limb.
That's great to know. Thanks, Johannes. Unfortunately, that's all we have time for today. There are so many more questions. If you'd like to learn more about Johannes's lab, you can find his details on the University of New South Wales website. You can also visit the Cellular Agriculture Australia website for more information on how to get involved in the field, more free events and conferences. You can join our public Slack channel, and there's lots of useful learning materials for beginners as well as experts in the field. And the link to that is www.cellularagricultureaustralia.org. Very easy to remember. And the easiest way to stay up to date with how the field is progressing in Australia and worldwide is through signing up to our bi-monthly newsletter. And I promise we won't spam you. You can subscribe to that on our website. So thanks so much, Johannes, and I'll pass it back to Luan.
Thank you.
Thank you, Bianca. Thank you, Johannes. Well, that really brings us to an end of another Convergence Science Network event. And I hope you found Johannes's talk informative, inspiring, and hopefully it will have you thinking about the issue some more. And please also visit the website, convergencesciencenetwork.org.au, to subscribe and make sure that you're kept up to date with other events and news that are coming around to us. Thank you very much, Johannes. You've certainly informed us, raised some very interesting issues, and really shown a pathway for an exciting development where the sciences are going to hopefully lead to a new and successful industry in Australia. A special thank you to Bianca Lay, of course, for her support in making this event possible, for her moderation. Thank you, Bianca. Wishing you and great success with Cellular Agriculture Australia. A big shout out to the Convergence Science Network sponsors who make these events possible, and we offer them to the public at no cost. Stay healthy, be well. Thank you and good evening.
Thank you.